Oh, welcome everybody. Hope your breakout sessions were great. And so now you're in a lightning session on biobanking efforts and resource supporting exposomics research. So it's a little bit of a turn from what we've had all morning. And so far our sessions have explored the concepts, approaches, and technological innovations that are shaping the future of exposomics, from influencing policy and advancing the Human Exposome Project to harnessing big data, AI, and other bold initiatives. Yet all of these exciting possibilities hinge on one critical foundation, and that's access to high-quality data. So to truly measure the exposome, we want to turn these ideas into reality. We need robust biological samples and biobanking resources, and without them, even the most innovative strategies are going to remain theoretical. So this session is going to focus on a set of biobanking efforts and resources that have the potential to make exposomic research possible. Each researcher will introduce themselves, and then in 1 or 2 slides, they'll provide you with a snapshot of their cohorts, presenting the key aspects related to exposomic research that can be supported by their biobank. Then we can open it up, if there's time remaining, to the group so they can ask questions about how these resources are being developed, what challenges remain, and how they can support the transformative work we all envision. We can dive into the practical side of exposomics and explore how we can build the data infrastructure necessary for real scientific impact. So, we'll have our first cohort lightning slide. All right. So Alicia is online. Please introduce yourself, Alicia, and then take it away with your slide.
Great. Thank you so much. It's a real delight and privilege to be here. I'm sorry, I wish I could be there in person, but unfortunately, this week has been a little crazy and does not permit that. So can you see my slide okay?
Yes.
Okay, wonderful. So I'm an assistant professor at Mass General Hospital in the Broad in Boston, and I'm a geneticist by training, but I've been very interested in exposomics, particularly as of late, because I'm especially interested in health disparities. And differences across populations are not fundamentally typically driven by genetics, or at least genetics cannot fundamentally explain why there are such differences in prevalence of health across populations. And so to try to get to the bottom of some of these, I've been heavily involved in the Global Biobank Meta-Analysis Initiative. This brings together more than 24 biobanks across more than 2.5 million individuals around the world. Multiple of these biobanks actually have high throughput proteomic profiling data. So, DNA makes RNA, makes protein, and this is sitting most proximally to disease state and therefore makes really excellent biomarkers for disease status. And so proteomic data has become increasingly available through cutting edge advances in technology development. So there are tens of thousands of individuals with proteomic profiling on thousands of proteins in the UK Biobank. There's also thousands of individuals in FinGen and tens of thousands of individuals being profiled in all of us as well. This proteomic profiling data is highly, highly predictive of disease risk. For example, areas under the curve, a common measure of how accurately biomarkers are working to predict common complex diseases, are routinely over, say, 0.8, for example, in type 2 diabetes. But the question is, why are they so predictive? To understand our biomarkers, we need to understand whether they're causal or what they're relating to. So in fact, only about 8 to 15% of proteins are causally associated with disease. And in fact, the vast majority of the variance in protein levels relates to lifestyle risk factors like smoking and drinking, which are a battering ram to the proteome. More than 50% of proteins have major associations with both smoking and alcohol. So this is amazing because it gives us an objective, quantitative measure of lifestyle. It's like a gene chip for the environment. It gives us something that we can replicate across biobanks. These are also specific to the environment. So for example, a protein smoking score is not predictive of alcohol use and vice versa, even though these are helping explain disease risk. With that, thanks so much and happy to turn it over to the next speaker.
Hi, I'm the next speaker. So you've already been introduced to me, Susan Teitelbaum. I'm an environmental epidemiologist at the Icahn School of Medicine, Mount Sinai in New York City. And I am the director of the Human Health Exposure resource data center. So I wanted to tell you a little bit about HERE, the program, and also HERE, the data repository. So HERE was envisioned by NIEHS as a way to provide researchers who had existing cohort data, who may never have thought about incorporating environmental exposure into their research, with a way that they could. And so HERE is made up of a network of laboratories that can provide measurement of environmental biomarker data, both on a targeted level and untargeted level. And we also have a data center, which I run, which provided statistical analysis with advanced statistical abilities to look at the results of the biomarkers in relation to the cohort data and use methods in order to get an exposomic view of how associations may be with health outcomes. And then we had a coordinating center that sort of kept everything else running. So I'm going to focus on the HERE data center. We provide a large de-identified data repository of epidemiologic and environmental biomarker data, and it's downloadable by the public. It's a public repository. The data once has, it goes under embargo for the PI to have an opportunity to use it for a year after the labs have generated the research, their results, and then it becomes part of the repository, and it's available to anybody who's affiliated with an institution that has an IRB or an ethical review board for European institutions. And the data can be downloaded and used by the individual who downloaded for their own research purposes. We also have, and those are, you can download individual studies, but we can also provide harmonized data sets from, right now there's 55 studies in the system. Not all of them are off embargo yet. And that encompasses over 35,000 individual subjects. And we've had over almost 65,000 specimens measured for either targeted and or untargeted biomarkers in the repository. And the untargeted labs have the ability to measure over 236 chemicals. So the graph sort of gives you a real high-level picture of the exposomic nature of the HERE data repository. So each of these different health outcomes, because we have a wide variety of health outcomes among the cohorts that are in the repository, and what we're showing you in the bar length is how many biomarker measurements have been made for each of those particular health outcomes, and the colors represent the various chemical families that have been measured for each of those outcomes. And one of the things I want to point out specifically for sort of an exposomic view are the sort of green, the light green, they tend to be at the end of the bar. Those are all untargeted measurements that have been made in relation to every single one of these health outcomes. So we've incorporated a lot of information for all of these health outcomes. So if you scan the QR code, it will take you to the HERE Data Center website where you can see this on the front page. And you can also look at the metadata of all the studies. And if you'd like, and you haven't already, you can register for access to the data, for all the data that's in the repository that is off embargo, and start exploring and start looking at this data however you want to look at it, probably way beyond the original specific aims of the studies that generated this data. Thank you.
So thank you very much. My name is Roel Vermeulen, Utrecht University in the Netherlands, and I'm representing here the European Human Exposome Network project or program that was a program of 100 million sponsoring 9 projects in Europe. And I'm going to show you here the data tools that are available for everyone to use, which is currently being picked up by the International Human Exposome Network to make all this information fair. So the easiest is actually to go to the URL that's actually listed here, which is the Exposome Toolbox. That actually was the final product of all these projects. This is hosted under the Elixir community. There's a new Exposome community, so everybody can actually upload their information about tools that are available. The most important resources that I wanted to highlight here in this session is the European Health Research Data and Sample Catalog. We'll show you an example of that, what is currently in there and how that works. The Exposome Maps, which is all the unified geospatial information across Europe at a very high resolution, and we'll show you the examples as well. Just wanted to highlight also the Exposome Explorer database, which is run by IARC, which actually summarizes all the UN biomonitoring data from data sets, but also curated from the literature. An incredible resource if you want to actually project your results onto the results of other studies. There's also a whole dedicated area for statistical resources for how to analyze this information. Just to give you a high-level overview of the data catalog, so at the moment we have 72 studies within the data catalog, comprising more than 4 million individuals within those cohorts. That resembles about 200 million biological samples that are within those studies. And most of these studies are longitudinal, so really following a more life course perspective. What is important here is that the 72 cohorts map to 10 networks or consortia. And why this is important is because those consortia have worked together across different cohorts. And that means that not only the data is fair, but also the harmonization protocols. So if you want to map your cohort towards one of these networks to actually compare your results or do a pooled analysis with these resources, that is possible. So you can express your data as being part of the athlete consortia, of being part of the Exposome consortia, using the same harmonization protocols, which are all available. At this moment, there's more than 2,000 variables that have been harmonized across these cohorts, so that resource is completely available, and I would invite you to go to the BioTools and actually look into the catalog. It goes a little bit further because this is actually the data within cohorts, but within Europe we now also have harmonized some of the census data and the exposome data, covering more than 25 million health records at this moment. This is just an overview, which I know is difficult to read, but this is actually from one of the networks. This is from the HEDEMET study that is also represented here. And what you can see are the different cohorts. What is important to take away is that these different cohorts resemble different stages of the life course, so that actually enables us to track the exposures and health impacts across the different life course. You can see all the different columns, which indicates basically that there is proteomics data, metabolomics data, or epigenetics data within these cohorts available to actually look at these biological imprints. So again, I would invite you to go to the catalog and actually see what data is there and if actually that would suit your research purpose. Just to also show you that we now have more than 33 environmental factors completely harmonized across Europe at a resolution of less than 100 meter by 100 meter, time varying from the year 2000 to the year 2020. All this information can be found on the Exposome data platform and is open for everybody to use and link their information to. There's also a linkage tool, which is GDPR proof, so that there's no exchange of geocoordinates with our site, so that you actually can retrieve this data without actually identifying your people within the study. I'm just showing you this beautiful picture, but it's just showing the thing that we now can do within the European population, so that's really looking at all countries at a very fine grain and also looking over time, and this is just comparing 2000 with 2020. Open circles is where the environment improved for these different countries. These solid circles is where actually the environmental circumstances became worse within Europe, mostly temperature, that's obvious, but also still impervious surfaces. And it allows us basically to track the environmental state in Europe and link this all to cohort data and see what the impact is. And with that, I just wanted to finish this as the resource within EHEN, and again, just go to the URL and see what resource is there. Thanks.
So hi, thanks again for being here. I'm Denis Sarigiannis. I'm chairman of the board of the and director of the National Research Foundation of Greece. And I'm now representing the follow-up, let's say the sequel to EHEN, in a way, because the follow-up cluster of projects for a budget of about 50 to 60 million, I think, is called ExposomeHealthNet, and that has just started, actually. So what I'm showing here in a nutshell is the data and the data resource that's available both in terms of data and biobank samples in the project that I'm leading, the Exposome Project for the environmental exposome. We're focusing primarily on let's say what you might call emerging stressors, like ultrafines, bioallergens, light, noise, but also hazardous waste, related contaminants, including, of course, carcinogens, but also more subtle compounds in terms of, and the legacy compounds. So that's in terms of what we're trying to look at. And we actually avail of a number of biobank samples. We have, I think, a very large biobank, including, as you can tell, newborns, children, pregnant or postpartum women samples, adults, but also workers, and actually also a pretty large series of environmental samples from across different countries. What you see here is just the data and the map for the Exposome Project. We have 21 cohorts for more than, I think, 10,000 samples just for our project. So if you multiply that by about 6, which is the other data that's available, the other samples that's there, we actually get a pretty large resource. What we have is also, as you can, in the biobank samples, of course, untargeted and targeted exposomics as analytical, let's say, exposomics, but also metabolomics data and mRNA, so either with DNA, with microarrays, or depending on the time and of the project, but also or with RNA-seq transcriptomics data to actually derive what we think is the thing that is missing, and that is to try to draw the biological link between the gene expression, the actual exposure data, the gene expression data, and then eventually the biological data that's closest to the phenotype, and that's of course metabolic perturbations. We also have proteomic data for a number of these samples, not for all of them, because our thinking is that you first do an untargeted approach between transcriptomics and metabolomics. You do the pathway analysis and joint pathway analysis, and then you corroborate what you find as suspect adverse outcome pathways or networks with targeted multi-omics, including in this case then proteomics. That's to try to be more efficient and enhance the usability of the data. So all of these data sets actually currently available. We have built since 2013 at the HEALS project, one of that at the time the largest European project on the exposome, with a budget of 15 million euro, a resource, an exposome data management system that was actually pulling together a lot of data sets, including, as you see here, also information on environmental condition state. This is, you see the different types of data sets we're using here, including not only air and water, but also soil characterization and indeed distribution of a number of key stressors. But also combined with, as I said before, noise in particular, and also light, artificial light mapping. Now, the data come not only from regular monitoring systems, regulatory monitoring systems, but also from personal exposure information, using wearables, using different types of Internet of Things kind of sensors. So that's why you have a very significant granularity in terms of the spatial and also temporal resolution. I think that's also very important in an exposome sense. So this resource is actually also following the FAIR and open science paradigm, especially at the National Research Foundation of Greece. We're one of the two leading organizations in the country on the European Science Cloud, Open Science Cloud, sorry. And so the data is available for use, but you actually have to register your project and then try to get access to them to do further downstream analysis. Again, what I think, just a closing word on this, is that it's not just about data and biobanks and so samples to analyze. It's really about what you do with this data and samples. And so I think it is very important to combine in terms of, by way of resource for this community, not just the data and the samples, but actually the models, the algorithms that could help in deciphering what this very complex labyrinth of data actually could tell us. And I think it could tell us different stories, very important stories, which may also be different stories and with significance that varies according to the specificity of the actual location and type of and socioeconomic conditions of the population at hand. So that's it. I don't want to take too much time. Thank you.
Hello, I'm Jana Dutka. I'm the program director for the Genome Centers at the NIH All of Us Research Program. And up here is a lot of information, but I'm just going to run through it quickly. So we have currently available what we're calling our CDRV8, which was just released in February, and you can see that as part of that we have survey responses, physical measurements, genotyping arrays, short-read whole genome sequencing, electronic health records, Fitbit, long-read, a large amount of data. What is important is that not every participant that we have available, so we have over 600,000 survey responses, not every participant has all of the data available within the system. So you can see within our CDRV8, as you drill down to who has overlapping physical measurements, that number drops to 400,000. So you get electronic health records down to a little above 300. As you're adding in who might have Fitbit in there, then you're down to a little over 40K. So there's a large wealth of data, but depending on what you're trying to investigate and what sort of exposures you're looking at, that will determine your data set. We do have a large anticipated growth in our data set for 2026. We're planning to add several different types of data that were not previously available, including a large amount of methylation from long-read data, some bulk RNA sequencing, proteomics, hexed environmental linkages, and metabolomics data. Our biospecimens, as of April of this year, we have biospecimens on over 600,000 participants. These are all adult participants. While we have a pilot of pediatrics, we don't have a large amount of pediatric participants available yet. And these are the types of samples that we have available from participants. Some participants contributed blood and all the different biospecimen types. Some participants only contributed saliva. So again, what we have available on an individual participant is variable. We do have some internal projects that are currently utilizing our biospecimens in order to investigate exposures. And they're doing them in three very different ways. One is utilizing our participants as a resource. So they are actively recruiting into the All of Us program. They're performing their own research project, and this is the Nutrition for Precision Health. So these participants are actively recruited both to All of Us and Precision Health and the Nutrition Precision Health Project, and then they are followed either for a self-report diet or they are given particular dietary challenges, and then additional measures are collected for additional assays on them. We also have a partnership with NIEHS in which we are conducting an environmental health and exposomics study. And in this case, they're accessing our participant data and our biospecimens in order to perform untargeted metabolomics using the HERE resource, one of the HERE resources that you heard about earlier. And this is also combining with our data to do a broader look at the exposomics investigation that you can do within our database, and particularly focusing on diabetes. We also have a multi-omics pilot study that is specifically focused on using our specimens to generate a large amount of multi-omics data. The pilot study is mainly focused on data generation, and what then other groups who would come in and utilize this data could be applied for exposomic studies. Our data types we expect to continue to expand, so I do not have a comprehensive list of all the data types that we will at some point have available up here. This is just what we have fairly certain we will have in the next year and what we have right now. And we are working currently on developing a way for broader access to biospecimens. As I said, all of the projects currently are either internal to the consortium or partnerships within NIH, but we are working on a broader access mechanism that we hope to roll out in the near future.
We run out of the time, so I will talk from here. Hello, everyone. My name is Bamai, Associate Professor at Center for Environmental Health Science, so-called, in short, CEHS at Hokkaido University. I am an environmental epidemiologist specializing in exposure science, including operating birth cohort studies and assessment of environmental chemical exposure. As for cohort study in Japan, to my knowledge, there are currently over 70 studies in total. These include cohort focusing on adult and elderly people with specific disease and general population based on the regional based on general population. Of course, I cannot introduce all in just 2 minutes. So today I've prepared a slide focusing on the birth cohort study because I am working on birth cohort in Japan. So maybe you have heard about the CEHS. Also, Gary this morning introduced the Nexus. So CEHS were already included in the part of this program. So I am involved in the, especially for the Hokkaido study and CEHS, so-called Japan Environment and Children Study. So inspired by Gary's speech this morning, this one is not the final slide. So I updated this morning, so it's okay, I will skip that part. So what distinguishes birth cohort most is the rich information collected during pregnancy, descriptive data, biological data, and biospecimens, which allowed us to examine impact on the next generation. As you can see from the slide, nearly all cohorts collect and bank biological samples during pregnancy, such as urine and blood. At birth, samples like cord blood, umbilical cord tissues, and breast milk are collected. After birth, biospecimens including urine, blood, saliva, baby teeth, stool, and cell are collected from the children. In terms of environmental exposure, many cohorts focusing on environmental chemicals, indoor dust, or air sample for internal and external exposure assessment. Some are not focused on chemical or pollutant exposure. However, they are more focused on maternal mental health during or after pregnancy as an exposure factor. As for outcomes, most cohorts investigate low birth weight, and also SGA, allergic disease, developmental outcomes, neurodevelopmental disorders such as ADHD and ASDs, and increasingly childhood obesity, which is also becoming a more concerned development in Japanese children. In the Hokkaido study and the CEHS study, we are collecting pivotal development, and in the Hokkaido study, we have also begun collecting data related to reproductive health outcome and also fertility for the next generation. Regarding international data sharing, sorry, this one is, I added the one column in the last. Maybe you can receive from after the symposium. Yes, so I added the information for data sharing. Currently, Hokkaido study, and I believe Tohoku study TSCD are sharing data under certain condition. Other cohorts are currently under consideration. Lastly, also, although I haven't touched with the sample size yet, I want to highlight that the 3 cohorts, Hokkaido and Czech and TMM, those cohorts have collected data from 20,000 to over 100,000 participants. So importantly, all 3 are either tracking participants from the fetal period into adulthood or have planned to do so. That concludes my overview of birth cohort study in Japan. Thank you very much.
So hello everyone, I'm Poornima Prabhakaran, physician and epidemiologist by training. Been working in public health for nearly 2 decades in India. I direct a Center for Health Analytics Research at Ashoka University, but I'm also affiliated with the Center for Chronic Disease Control, an organization which turns 25 this year, where I head the portfolio of environmental health work. So in the spirit of this being a lightning session, we were asked to hold 1 slide. So it's a tall order to even start to speak about the resources in a country like India. It's countries within a country. So what I'm going to try to do here is actually just give you a snapshot of resources that we have in our own organization. And I'll start with the CARS cohort. It's one of the longitudinal studies in India that was established way back in 2010. We like to call it the Indian Framingham, only it's 3 times the size of Framingham. And it's a cardiometabolic risk reduction surveillance study established in 2010, but with several rounds of data collection over the years, we continue to do it. And it's intensively phenotyped. It was set up in 2 cities, Delhi in the north and Chennai in the south, for those who are familiar, and established primarily for cardiometabolic risk surveillance. But over time, this has served as a platform for research across different domains, and I'll come to that in a minute. I just want to describe the other cohort, which is in 2 other geographies in India. This was set up for prevention of diabetes and hypertension, again focusing on cardiometabolic disease. This has a sample size of about 12,000 across the 2 cities. I've listed here a few other resources, again, across different geographies in India, which allows us to study the different vulnerabilities across these regions. We have the Solan study, which is in Himachal, a very hilly terrain, which is also to look at the cardiovascular disease profile in this region. And the studies that are listed after actually have become part of our consortium within our GeoHealth Hub. Now, the GeoHealth Hub was funded in 2016 by NIH. It is one of 7 in the world. And we decided to use our longitudinal cohort study to leverage and look at air pollution. This was a very topical issue in India for us in that time. And what we have managed to do is that beyond the biorepository that we built for this cohort study, which is over 1 million aliquots of samples, which includes whole blood, plasma, Buffy coats, and samples for methylation studies, we in the GeoHealth Hub have actually established an environmental exposure assessment for air pollution and temperature. And this is a very unique model, which is called an ensemble model, which takes into account whatever data you can get from ground monitoring stations across the country, but also satellite data, road networks, fire counts of the positions of power plants. And this ensemble model has allowed us to build a spatio-temporal model for the entire country at a very fine resolution of 1 square kilometer. This is from 2008. up to 2022. We are updating it further and expanding it beyond the culprit pollutant PM2.5 to NO2, SO2, and ozone, as well as ambient temperature. So now we have this environmental exposome for the entire country at this very granular resolution, and we leveraging our cohort and all the biomarkers and outcomes that we have started to establish the associations for environmental exposures and health outcomes. And somebody mentioned in one of the earlier sessions today how difficult it is to establish a cohort and follow it up across your lifetime. So in the second round of funding that we got at our GeoHealth Hub in 2021, what we did was brought into our consortium all these cohorts that I have mentioned below. These are what we call following the principle of interdigitating cohorts. So using the principle of a life course approach, we have assembled across the lifespan pregnancy cohorts, child cohorts, adolescents, young adults, the older adults that we had in our own cohorts, as well as the elderly. So bringing together consortia partners, we are going to be able to kind of leverage the environmental exposure assessments that we have built within our own portfolio of work, but also partnering with other researchers. We have assembled a life course of health outcomes. And what we're trying to do within this round of our funding in the GeoHealth Hub is to kind of examine exposure to environmental factors, a very topical issue for our country, but look at a range of health outcomes across the life course. It's going to be very difficult to read all across those columns, but I just want to say that we have a huge biobank in India now, and we're looking to kind of mimic what they did with the UK biobank and open up this resource. Our environmental model itself is actually up on our own website at the Ashoka University. It's up, open for public health researchers, but also policymakers, and anybody else who wants to access the exposure assessment model that we have developed. It's also up on the Harvard Dataverse, given Harvard is our collaborator. I'll close there, but happy to share further resources if anybody is interested. Thank you.
Thank you very much. Good afternoon. I'm Jana Klánová from Masaryk University in Brno in the Czech Republic, and I'm also the coordinator of the European Research Infrastructure on Human Exposome Research. But today I am not here wearing the IRIN hat, but wearing the BBMRI hat. And before I start, I should explain what I'm talking about when I'm speaking about European infrastructures. Already some 15 years ago in Europe, it was established something we call European Strategic Forum for Research Infrastructure, which is the intergovernmental mechanism that is deciding on the priorities of the infrastructural capacities for excellent research. So it is long-term operating intergovernmental forum that is deciding on the future of the European science. And in the meantime, in those last 15 years, they already established about 40 infrastructures in different areas of research. And one of them is BBMRI, which is the research infrastructure for European biobanking and biomolecular resources. And I was hoping that Jens Habermann, as a Director General, will present today, but since he was unable to come, he asked me to present on his behalf. Originally, the BBMRI was established for coordination of the hospital biobanks. If you want to do the research on rare diseases, you really need to have an access to the samples across different countries. But gradually, they developed the capacities not only in biobanking and having the records of existing samples, but also the related data. So they now are developing the resources for genomic, proteomics, metabolomics, and others. And this is also the research that is important for not only for the biomolecular, but also the exposome research. And of course, they do not run the long-term population cohorts, but they happily collaborate with a number of the European cohorts, providing them the support. And they also coordinated one of this cluster of the EHEN exposome cohorts that Roel spoke about. and they focused majorly on developing the IT tools for organizing the data, looking into the data, and interpreting the various data. How do I move on? So this is briefly about the BBMRI, and I said that BBMRI is providing the support also to many European cohorts. And you already heard from Roel and from Denis that there are multiple European cohorts or many generations, but I think one of the things that Europe can be proud of are the European birth cohorts, but they are scattered all over. There are many in different countries. And in my second slide, I would like to present the work of Martine Reihai from IS Global, because she's been working in the last 15 years on trying to organize the European birth cohort. So in the series of the follow-up projects, she was really working with different birth cohort, trying to bring them together on the same platform, use the same protocols, use the same metadata schemes to really make the data comparable. And in that, she also collaborates with the BBMRI. So there are resources like the Molchanis, which is really the software that helps to register the European cohorts, including their metadata, but also other softwares like Armadillo or a Data Shield that is built to allow for the federated approaches. So currently, she just finalized the Athlete project, which is again one of the EHEN cluster that invested a lot of effort into federated approach to access to various data from the European cohorts. So those are just few examples, and I said at the beginning that I am coordinating the new European infrastructure on the Human Exposome, and all those discussions that we are having here about what are the key elements, this is something that we've been discussing in the last 5 years. What are the key elements of such infrastructure? And cohorts are definitely one of them. So I would be very happy to discuss across different continents, you know, the initiatives in Japan and South America and Africa, how we can bring those resources together. Because I should say that the whole S3 concept is not Europe-centric. You know, those cohorts or those research infrastructures are initiated in Europe, but many of them are really reaching out to other continents. Many of them are global, and we hope that IRIN will be really global infrastructure, and we will come the collaboration with the nodes in different countries. Thank you very much.